Hi, well, I think we'll get started on time so that we can give our speaker a chance to tell his whole story. My name is Rick Cody. I'd just like to give you a few opening remarks about the Personal Genomic Medicine Sidore series that we're concluding with this lecture today. As you know, the Sidore series is a wonderful opportunity for the university to bring in on different thematic topics, very interesting topics for us to consider over the course of the year. And I'm not going to read that slide, but it's really a really exciting opportunity to bring the entire university community together. Some of the previous Sidore topics include the following up here. And you can see that the personal genomic medicine that we're offering this year is a distinct departure from many of the more liberal arts-oriented ones. It's been a very exciting series that we've had. And just to summarize what came before, for those of you who may have missed one or more of the topics that were presented before, we started off the series with Jane Foster giving the first lecture with a compelling description of how the microbial communities that live inside us can influence not just digestion, but other aspects of behavior, the immune system, endocrine function, etc. We followed that with Michael Snyder, who came and talked to us about insights into how various omic technologies, not just genomics, but other kinds of high data, big data type of approaches, can generate an integrated personal omics profile to assess disease risk, monitor disease states, and enhance therapeutic treatments. We followed that one with Barbara Koenig, who addressed the ethical, social, and legal implications and responsibilities that accompany acquiring, interpreting, and sharing these vast genomic data sets. And finally, the talk that preceded this one, Elaine Ostrander's talk, provided insight into human health and disease that can be obtained by studying the genomics of naturally occurring dog populations, as well as appealing to dog lovers and breeders. I wanted to mention that aside from the first one, we have videotaped these, and we are videotaping today's as well, and they're available on the Sidore website. Without further ado, I'm going to introduce Dave Poshetsky to say a few remarks about today's speaker. Thank you for coming.
So we're really excited to have John Hawks here to close out the Sidore lecture series. This whole series has been sort of introspective. How do these modern genomics approaches inform us about our own existence? And I guess we come to the most profound question of them all regarding our own existence with the story of our own evolution. And so John is a real leader in this area, and we're really glad to have him. He's a full professor at the University of Wisconsin. Prior to that, he did his PhD at University of Michigan with a postdoc at Utah, I believe, correct? And so John, again, is a leader in the area of paleoanthropology and also paleogenomics. And today he'll talk about some of that stuff. So thanks again, John.
Thanks. All righty, I'm going to see if I can't get everything rigged here. And we'll hit present. And this may rattle around a bit. Okay, so this is an incredibly rapidly moving field, ancient genomics. And the field that I'm going to describe to you literally did not exist in this way 5 years ago. So it is now, just really changing so fast that it's very difficult even to summarize what we're working on. My work is really in a very small part of this area. I work on how to integrate these findings from ancient DNA into our traditional models of human evolution, and what parts of this are really new, what parts are reflecting new information that we have never really thought about before versus solving old problems, many of which we've been thinking about for 150 years. So I'm going to focus on some of that historical background. In the course of that, I'll be describing a little bit of DNA and analysis that's been conducted by my own research group, but a lot of DNA and analysis that's been contributed by research groups from around the world. And I want to point out, every time I give a talk about this, I just have to indicate the amount of effort and expertise that has gone into developing, first of all, the technology to do ancient DNA sequencing, which was developed in large part specifically for these early hominin contexts, and now is being engaged even more broadly to look at the evolution of other species and ecologies. So this was really developed first for humans. It was developed, leaders in this area are Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology, his postdocs Matthias Meyer, Ed Green, Johannes Krause, who's now has got his own research group at the Max Planck in Tübingen. There's an incredible group in Denmark, Eske Willerslev and his group, that have been engaged in finding information about more recent hominin populations. All of this stuff goes in. And it's the work really of an entire community of people, a community of people that did not exist in this way 10 years ago, certainly. And so this is, I'll tell you guys, I gave a lecture at our annual physical anthropology meetings last week. And I was there in a session with people who were specialists in ancient genomics, and they all gave their talks. And I got up at the end and I said, I don't know really how to feel about this because it's never happened to me before. I am the oldest person presenting in this, right? And I'm not that old, right? So it's a field that has really undergone a very rapid transition. This is Mount Carmel. It's on the eastern end of the Mediterranean, the Levant side, where the sun is rising, Levant. It's a very famous site historically, biblically. It's also a famous site in terms of anthropology. The caves in Mount Carmel contain some of the earliest evidence of modern humans anywhere in the world, and in particular, the earliest evidence of modern humans outside of Africa. This is the Rock of Gibraltar. Gibraltar is a place, it's beautiful. I've been privileged to spend a good deal of time there not because of the sunshine, although that's very nice, but because of these caves, which are full of archaeological remains left by Neanderthals more than 30,000 years ago. Gibraltar is the place that we now presently think the last Neanderthals that we've ever discovered lived. They lived just over 30,000 years ago and left their traces in these caves for us to find archaeologically. So one end of the Mediterranean, the east end, to the other end, Gibraltar, you've got this incredible range of populations. That is a very tiny fraction of the world, but it's only about half the total extent of range of Neanderthals during their existence. So I'll show you here, the Levant has Neanderthal remains along with those earliest modern humans. We've got Neanderthals here in the Caucasus. We've got Neanderthals across southern and northern Europe into Spain. So Neanderthal existence was across this area. But the age of genomics has shown us that bone fragments from sites well into Central Asia contain a similar signature as the genetics of these Western European sites. And so Neanderthals as a population, we now appreciate as a population that stretched literally from the Altai, from Central Asia, all the way through Western Eurasia. That being said, I'm describing to you a population that we now know from genetics and that we previously thought from their lightness on the landscape was extraordinarily thin on the ground. That's my friend Steve Churchill's book, Thin on the Ground. It's a great description. Across this entire range, at any given time, from the, now we understand, more than 400,000 years ago that Neanderthals originated, up to 30,000 years ago when the last Neanderthals lived, there were probably never enough Neanderthals to fill a good college football stadium in the US. They were a very, very sparse population. The world was a very different place when they lived in it. Every one of these sites that I've highlighted here is a site that's given us some genetic evidence from Neanderthals. Most of this genetic evidence is in the form of mitochondrial DNA sequences. I know a lot of you know something about genetics and know about mitochondrial DNA. Just briefly, it's a very small loop of about 16,000 base pairs of DNA that exists outside of the cell's nucleus and is inherited along with the cytoplasm through the maternal line. So the mitochondria in the egg remain along with you. The mitochondria in the sperm are actually ejected at the time that eggs are fertilized. And so it's inherited along the maternal line. That gives us only one piece of the evolutionary trajectory. In 1996, when the first mitochondrial DNA sequence was published from Neanderthals, it was clearly quite different from the mitochondrial DNAs that had been found for all humans living in the world today. And that data only continued to accumulate. Right? We've now looked at hundreds and hundreds of thousands of people's mitochondrial DNAs. We've never found one like the Neanderthal sequences that we have here. And so in 2010, when the first nuclear genetic evidence of Neanderthals was published, it came as a surprise to many people that actually humans around the world have a legacy of this Neanderthal nuclear genetic contribution, one that marks up to around 2 to 3% of the genomes of everyone living outside of Africa now. And that's an incredible finding, that this ancient population that once existed, it's now gone, actually still persists within our genomes today. If you consider that, for many of you in the room, around 2 to 3% of your genetics comes from Neanderthals. This is true of more than 6 billion people in the world today now. Everyone outside of sub-Saharan Africa. Sub-Saharan African peoples have a trace of Neanderthal DNA, but not very much of it. Outside of sub-Saharan Africa, more than 6 billion people are more than 2% Neanderthal. That's like having 200 million Neanderthals walking around the world. That is a lot of Neanderthal, right? Neanderthals today in extinction are more successful than they ever were when they lived. And that's the course of our evolutionary history, right? Becoming part of the modern human expansion was actually the way that we have preserved the DNA of many ancient populations of people, most of which, except for the Neanderthals, we did not know existed before 5 years ago. So let me give you some perspective on this stuff. A, because I'm a Neanderthal nerd. I really love these guys. And B, because it's helpful to go through a bit of history to help people understand that the sort of questions that we're asking today about Neanderthals and their genetics and their biology are questions that were not possible to conceive of before 10 years ago. Really, we were not thinking about things like Neanderthal immune systems, Neanderthal digestion, those sorts of things that are incredibly important in evolutionary terms. All that we had were these bones. And we've got a lot of Neanderthal bones, I got to tell you, right? This is one of the first to be identified. This is the Gibraltar Neanderthal skull from Forbes Quarry. Forbes Quarry was an active quarry that no longer exists. It's now, the site's gone. It was quarried out. And that's true of a lot of Neanderthal sites. This skull, found in 1848, was seen by this man, Charles Darwin, at Down House in Darwin's study. You can sort of imagine him sort of in his looking glass here, putting the skull underneath of it and looking at it. And Darwin's been closer to this one than I have, as it turns out. I've seen lots of Neanderthals, but I haven't been to the British Museum where this one is to study it. This is the original Neanderthal from Feldhofer Cave in Germany. It's from a place very near Dusseldorf called the Neanderthal, which means the Neander Valley. And Neander was the Greekified name of a guy named Neumann who lived in this valley. The valley became named for him, and so it's the Neander Valley, the New Man Valley. And in English, this came in before Germans changed their spelling. And so in German today, this is spelled Neanderthal without an H. But in the 19th century, 1856, when this was found, it was Neanderthal with an H, pronounced the same way. It comes into English and it becomes Neanderthal. And so Neanderthal is how it's come to us. It's part of a partial skeleton from this beautiful place. People used to picnic there. Here's the cave, the Feldhofer Cave, which became an industrial quarry to build Dusseldorf, and all of this is no longer there. In fact, if you go to the site now, they have rehabilitated this. The first time I visited this site, there was a chain link fence around it and a gravel sort of parking lot surrounded by junkyard cars and actual junkyard dogs barking at you. It was like an old industrial site. You couldn't go anywhere near it. There was a beautiful museum across the road to commemorate it, but the site was just totally junk. They cleaned that up. They've rehabilitated it. In the course of doing that, they did some minor excavation in the gravel parking lot and found additional pieces of the original skeleton, along with pieces of a second individual, which was identified as another individual from its mitochondrial sequence. In this park, you see these little crosses? I love these, because on one of them, in a typical sort of German monumental way, you've got the original 300 base pair hypervariable sequence from the mitochondrial DNA that was this individual, which was the first mitochondrial DNA sequence from Neanderthals. They've got it there, A, C, T, C, G, T, etc., on this concrete thing. The data that we have now from one high coverage Neanderthal genome, not from this site, from the Altai, the data that we have now would require a monument 10 kilometers across. The kind of data that we're working with ancient DNA now is in some instances as high quality as we can get from living people. We know more about Neanderthals' genetics than we know about many human populations' genetics, which is, on the one hand, a sad statement about our priorities in sequencing human diversity today, but on the other hand is an incredible testament to our ability to see into this prehistoric record that did not exist for us. It is totally new. Okay, so that's the history of Neanderthals, but you guys know Neanderthals from another context, and that is the Neanderthal context, right? When you say, man, Fred, Representative Fred from Congress, he is such a Neanderthal, that means something, right? It means progressive. It means that you're always looking out for the rights of women. It means that you're going to spend a lot on health care. No, it means the opposite of this, right? Neanderthal means stupid and backwards. And of course, during the course of our history of looking at them, 150 years of Neanderthals, we've been looking at them through a lens of them being the primitive, extinct, obsolete humans. Halfway to ape was the way that the first anthropologists encountered them. Now today we look at this and we say, wow, Neanderthals are a population that is as close to living people as it's possible to be and not be sort of counted among our number, right? They're human in brain size, they're human in most of their anatomy, they have just a few traits that are distinctive in them. They are not halfway to chimpanzee. They are really among the human family in most ways. But it has been a long course to rehabilitate them from this image of incredibly primitive or comical, get fast relief with Evo lotion, available in stone jar, of course, from the Geico commercials, right? Where it's the joke, right? Of course, so stupid a caveman could do it. And that image sticks with them. It has been my, sort of privilege in the course of my career to be in places where the Neanderthals walked and where other ancient peoples walked, and to think about how they lived, and in a way, to speak for them because they can't speak for themselves. And, until we discovered that actually they're not gone, they're still sort of inside of us, this was sort of like being the defense attorney for the guilty client. It's like you want to say nice things about people, but in reality, Neanderthals were not grossly mentally incompetent. They were incredibly insightful in their way. They lived a challenging life with means that most living people probably could not manage. And they exhibited sophistication that is under-recognized relative to other primitive peoples who lived at the same time. I love this chart because this is Ernst Haeckel, who famously drew phylogeny after phylogeny after phylogeny. And this one is a great one because here you have Homo sapiens at the top, as it usually is, and all the other anthropoid apes coming off the side of this, but the direct line that comes up to Homo sapiens. This is before any of these fossils had been discovered, except for Neanderthals. And so he had a place for the missing link. He called it Pithecanthropus, ape man, alalas, because Pithecanthropus was not going to be able to talk. He was just going to la 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 la. This sort of, that was really what Haeckel thought. But Neanderthals also were mute in Haeckel's point of view. He calls them Homo stupidus. And really that is sort of the template that the 19th century sets for understanding Neanderthals. In the 1980s, this skeleton was found from Kibara in Israel. It is Neanderthal in its affinities. You can see there's no skull there, but what it does have is a hyoid bone. And that hyoid bone, like the hyoids from this very early Neanderthal site, the Sima De Los Huesos in Spain, is identical in morphology to human hyoid bones. So the vocal channel of Neanderthals was like ours, different from chimpanzees. I'll mention that the Neanderthal FOXP2 sequence, one of these gene sequences that's been implicated as being important for language and development related to language, the gene sequence of FOXP2 of Neanderthals, same as ours, but there's some interesting stuff with FOXP2. I'll get to it in a little bit. This is a manganese dioxide nodule that is rubbed on because it was used as a crayon by Neanderthals to color things. This is a shell. This is the one side of the shell and the flip side, the opposite side of the shell. You can see on one side this shell has a natural band of color. On the other side it has red ochre pigmentation covering it in the same pattern. And these are shells that Neanderthals gathered and strung up. Bone tools that Neanderthals made. Cut marks on the distal wing bones of vulture, and these are quite common now. Neanderthals had a taste for wearing black feathers. We find birds that have their feathers systematically removed in multiple Neanderthal sites. We also find eagle talons that Neanderthals have strung and worn. And we know from statistical analysis, compared to the overall avian fauna, including large raptors, the Neanderthals seem to have been biased toward dark-colored ones. It's sort of like they were goth. We don't know really what that means, but we know that it's something stylistic. We know that Neanderthals were twisting string and making it. This is something that we thought was limited to modern humans until very recently. We know that Neanderthals were using space in caves in sophisticated ways, using specific areas for processing foods, for bedding down. We find their bedding material in some caves that has been buried over the years, but those sort of straw and things that are used for bedding leave opal phytoliths in the cave sediments, so you can tell that that was there. We find them transporting raw materials to make stone tools from, sometimes over hundreds and hundreds of kilometers. So they engage in rather extensive trading networks. So they weren't dumb, right? And I'll mention that in Gibraltar, in the base of Gorham's Cave, the cave I showed you earlier, we have found the first engraving made by Neanderthals. This one dating to sometime in Mousterian times, before 40,000 years ago. And you can see that we've come to call it the hashtag. It is what it is, right? But each of these incisions took a long time, deliberately sort of incising at to make. We don't know what this represents. It's at sort of a crossroads place in the cave, and some people think, well, maybe this is sort of like a diagram, a schematic of the cave, because it does sort of go this direction. Or maybe it's something else. We don't know what it is. We can't reach into their minds and see what they were thinking. But what we find is that the more that we look, the more that we notice this long tail of behaviors that were rare in Neanderthal times, that we don't find a lot of, but we've found multiple instances of. It's from comparing notes with each other that archaeologists started to notice the eagle talon thing. Wait, you found an eagle talon? Let me look at my eagle talons. Wait a minute. They're all wrapped around they've all been clearly altered at the bases to be strung up together. And we started to notice these things. It's by sifting through sediments that you see these traces of the fibers that were twined together to make string. We would never have found them otherwise. It's by finding new ways to analyze sediments that we find the opal phytolith traces of the Neanderthal bedding areas. And you start to get, at a microscopic level, a picture of behavioral complexity that was invisible to us looking otherwise. And of course, genetics is, in one sense, the ultimate manifestation of that. Looking at microscopic evidence, nanoscopic, invisible evidence from within bones that tell you reams of information. So this is my sort of home. This is the sample of Neanderthals from Kropina, which is the largest single assemblage of them. And I love this stuff. This is what I do. Some of you guys know I'm working in South Africa now on another sample, which is very much like this. Lots and lots of broken pieces of things. And getting information out of them has often been incredibly exciting for us, because it's from samples like this that you see variability, even though they're totally broken up. It's not like a sample of complete skeletons. Nonetheless, you're finding traces of what this population was like. But this bone, which is a little fragment of tibia from Vindija, I think I've got Vindija here, yes, Vindija in Croatia. It's very near Kropina, these two sites less than 10 kilometers apart, yielded the first nuclear genetic evidence of Neanderthals. And together with that bone, these two, today, each of them has given rise to about a 1.2x coverage Neanderthal genome sequence. These were the sequences first published in 2010. When these sequences came out and it became clear that, wow, the Neanderthal genome is quite different from ours in lots of ways, but it's less different from some people than others. This started to send us a message about what we were going to be able to find. Oh, do I want to say anything about this stuff? A lot of people have talked about red-headed Neanderthals, right? And maybe the redheads today they've got that from Neanderthals. I know a lot of you have heard this. It's probably not usually true. Today's redheads derive from many different initial founder mutations. Each of them has been selected in one mostly European, but in a couple Central Asian populations. And most of them are very recent. In fact, most of our pigmentation stuff today, light pigmentation genes, mostly less than 20,000 years old. These do not date back to Neanderthal times. But we have a couple that do, and it seems that Neanderthals had their own version of redheadedness that a couple of people today have. So we do have some heritage in pigmentation from Neanderthals. I used to give a similar talk and say, no, it's actually totally different. What you think is the same is not the same. It's mostly true. What you think is the same is not the same. When I talk to people about Neanderthal genetics and they ask me, you'd be surprised. I want to talk to you about my husband. You see, he has sort of a brow, and he's got hair on his back. And I think this is Neanderthal. I've always very carefully explained to people, that's not actually the same morphology we see in Neanderthals, right? The brow, for instance. pretty much humans today, if you see them having some sort of brow that looks prominent, maybe their forehead slopes backwards a bit, pretty much that's because of the development of the superciliary arches in the center of your head. Mostly, this is not the case that you have a big knob on the side of your brow that sticks out like this and is continuous across your brow in a Neanderthal way. Mostly, this is different morphology. And for those of us who are anthropologists who've thought about morphological evolution and how humans today might or might not have been connected to Neanderthals, we have these long arguments about whether this is really the same morphology or not. Are we looking at the same thing or different things? And I can tell you that mostly the things that you're going to look at people today that you think are Neanderthal derived are not. You look at somebody and says, that dude looks like a caveman. He's not. If you look at somebody and say, that woman looks like a caveman, I would look twice, right? Because that's non-stereotypical, and Neanderthal women did indeed have Neanderthal traits. And so, I look twice, but in fact, usually it's the case that it's not actually the same. But we now know that there are stuff that are the same, and it's mostly the stuff you don't expect. Okay, so this is David Reich, and David Reich is super, super smart. And one of the things I like most about him is he doesn't turn to super, super sophisticated things unless it's necessary. David has done a lot of analysis of Neanderthal genetics, and this is us together in the Altai. And David came up with what is really, really a very, very simple idea. The simple idea is, if we want to know whether a genome from the past has descendants in the present, the way to look at that is to compare different people in the present and ask a very simple question. Does one of them have more Neanderthal than the other? Because there are multiple ways that you and me could be like Neanderthals. It could be like the red-headed thing, right? Where I think that Neanderthals had this phenotype. I know that some people today have this phenotype. You have that phenotype. Maybe you're a Neanderthal descendant. Blood type is this way. Today, a lot of you, how many of you guys know that you're O blood? Blood type O, right? Some Neanderthals are blood type O. How many of you guys know that you're blood type A? All right, some Neanderthals are blood type A. That polymorphism existed. We haven't found a Neanderthal yet with B, but A and O at least are there, right? Does that tell you that if you're blood type O, you got that from an O-blooded Neanderthal? The answer to this is no, because in fact, when we look at O and its phylogeny in human populations, O is heterogeneous. There's lots of different types of O because O is a knockout mutation. It's a mutation that deactivates the ABO gene. And as a consequence of that, people who are blood type O actually have many different alleles. The most common of these alleles, which accounts for, I think, more than 90% of O blood people today, the most common of them, O1, originated, we know, 2.5 million years ago. 2.5 million years ago, there were blood type O things. Right? If you're blood type O today, it could be because you got it from Australopithecus. It's not necessarily that you got it from a Neanderthal. Neanderthals are your relatives, but it may be because you have some distant common ancestor and both of you inherited O from that. And likewise for A. So a particular gene doesn't necessarily tell you much about whether you have some Neanderthal ancestry. David's insight was to say, across the whole genome, actually these things average out. And if we only have genes in common from our distant common ancestors, then these two scenarios should be equally likely. It should be equally likely that you are like a Neanderthal for blood type, and I'm like a Neanderthal for hair color, or any other set of things. Over the whole genome, these should average out. What we in fact find is that when we compare people outside of sub-Saharan Africa with people in sub-Saharan Africa and Neanderthals, the ones outside of sub-Saharan Africa systematically look more Neanderthal-like. Across the whole genome, there's this bias, this skew. And that skew is a signature of mixture. populations outside of Africa have mixed with a Neanderthal-like population at some time in their evolution. A population that was already differentiated from the other ancestors of modern humans. This is a big data sort of approach. These are from the 1,000 genome samples. And this is just showing a straightforward chart that is the number of alleles in SNP genotypes across the genome that are shared with this Neanderthal sequence and are derived. So there's new things that aren't in chimpanzees, gorillas, etc. They're new in humans. Some humans have an ancestral allele shared with chimps and gorillas, etc. And some people have a mutation, a new allele, derived allele. And here's how many of those people share a derived allele with Neanderthals. For sub-Saharan African peoples, in this data set, it was around 667,000 or so. For Europeans, it's around 679,000 or so. And in East Asians, it's around 683 or so. This is, by and large, the signature of Neanderthal mixture in human populations. You'll notice immediately, Europeans have less than Chinese. This is puzzling. I have to tell you, this is really puzzling, right? Neanderthals didn't live in China. They lived in Europe, and Europeans have less. This tells us something important about our evolutionary history. Neanderthal genetics are sufficiently accurate as sort of a tracer of ancestry that you can use them to look at admixed populations and get pretty accurate views about where their ancestry came from. So these are African American people in the 1,000 genome sample, and you can see that some of them actually are mostly European. And many of them here are skewed a bit more toward the European side in terms of Neanderthal similarity than sub-Saharan African people are. That's a signature of their admixture after their immigration to the Americas. Likewise for Puerto Ricans. Puerto Ricans a mixture of European, Native American, and African ancestry. And you can see here they have a little less than Europeans do in terms of Neanderthal ancestry. So there it is. It's a marker of relationships. All right, I'm going to skip that stuff and go to this stuff. So that's all very cool. Then, also in 2010, something totally unexpected out of left field happened. The Max Planck group was working with Russian scientists from the Siberian branch of the Russian Academy of Sciences and sampling systematically bone fragments taken from archaeological sites in the Altai. The reason for this is twofold. One, actually this is a pretty unknown situation in terms of Neanderthals. Maybe we're going to find populations we didn't expect to find. But the other reason is that these caves are extraordinarily cold. Denisova Cave that I'm going to describe to you, Denisova Cave is year-round temperature 5 degrees Celsius in the sediments. So you're talking about in the deep freeze, really. You go into this cave and it's like a walk-in refrigerator. It's like being in the beer case at the grocery store. And so this is really an ideal preservation environment for ancient DNA. And in fact, from this one cave, we have today the most complete high-coverage Neanderthal genome, the genome from a toe bone, and the genome of another population that we didn't know to expect there. And when I show you this chart with all the zeros, this is because we've looked for admixture of this population with living people, and across most of Asia, it's only there at a bare trace, really a small fraction of 1% ancestry. But in Australia and Papua New Guinea, it's 5% of the ancestry of the people who live there. So you have this ancient population that we didn't know was there, that we didn't expect to find, that makes up 1 part in 20 of the genetics of these living people who are 6,000 miles away from the Altai. That's a puzzle. I've been fortunate to be a guest of the Russian scientists a number of times now at Denisova. It is a gorgeous place. It would remind you of Montana. And those of you who've heard of Siberia, you know it's very cold in the wintertime. You know that it gets to minus 40 and that sort of thing. And on the Siberian plain, it is in fact incredibly harsh. In the mountains here, this is the low altai, it's not at high altitude, but in the mountains, it is relatively less harsh. The winter temperatures sort of average a bit under minus 20 Celsius, so it's a little bit nicer. During the Ice Age, when the Neanderthals lived there, it was actually on average warmer than today. There was warmer season trees and so on are there, warmer season mammals are there. So this may have been not such a bad place to live. It's this bone, and I've shown a human one here next to it just to show you what we're looking at. We're looking at the distal phalanx of a pinky finger and just the base of it. And this line around it is the metaphysis. This is a growing finger. It's from somebody who's not yet adult. And from this bone, from around 400 milligrams of bone powder, has come a high coverage genome of a population that we did not know existed. We didn't know it existed because in terms of artifacts, this is what we've got, right? If we look at these artifacts and say, what's the difference between layer 11.2 and layer 11.3? There are some long, interesting blade-like tools, but aside from that, it's sort of Neanderthal looking, right? This is an archaic human population that's doing what Neanderthals do, more or less. And I would be honest with you, right? I would have expected it would be Neanderthals. We know that there were Neanderthals in the cave because we found them later. Okay, so let me sketch out, first of all, a big picture view. Because every time we get an ancient genome from anywhere in the world, we start to discover that there were mixtures that we didn't anticipate. So let me give you a sketch. Once upon a time, humans who ultimately, as modern humans, arose in Africa, and archaic humans, including Neanderthals and Denisovans, shared a common ancestor. That time seems now to have been approximately 500,000 years ago. And I'll show you why we think that in a second. After that time, those lineages evolved in partial isolation at least for hundreds of thousands of years until ultimately they mixed with each other. Denisovans mixed with the peoples that would ultimately give rise to East Asians and Oceanians, and a second mixture from Denisovans went into East Asians. Neanderthals mixed with the population that gave rise to all modern humans outside of Africa, and probably mixed a second time in order to account for the excess of Neanderthal ancestry in East Asia. I will add to this, once we knew what to look for, some very clever people working on African population genetics, including Joe Lachance, who's now at Georgia Tech University, went looking through African genomes for the signatures of population mixture. The concept here was, we knew before the Denisova genome came out that there was something funky about the genomes of Highland Papua New Guinea people. We had some genetic evidence from them. Looking at them, there was this sort of excess of ancientness across their genome. When the Denisova genome came out, you looked at that and said, oh, here's what explains that. They had this mixture from a population that was in fact very divergent from modern humans. And that accounts for this. So once you know what to look for, of course you can look at anybody's genome and ask, is there some excess of ancientness in it? Is there this shadow of a population that once existed? Geneticists have started to call these ghost populations. Populations whose existence you only know about because of the echoes of mixture that still exists in human populations. Lachance found evidence of one ancient African population contributing to Central African pygmies. Another research group found evidence for a second ancient African population contributing to southern African peoples. So you have within Africa multiple ancient populations which are mixing with each other to a slight degree, right? With Neanderthals, we're talking about 3% at most. With Denisovans, we're talking about 5% in a couple of populations, but a fraction of a percent elsewhere. With ancient African populations, we're talking about a few percent at most. And inside the genome of the Denisovans, there is a ghost population trace, something that's more ancient than Neanderthals are, something that contributed to them. We don't know what that is. It's a ghost population of Asia. So again and again and again, you've got these different populations that establish themselves geographically widespread, that existed for hundreds of thousands of years, and during some part of their evolutionary history mixed together to a small fraction. This is my chart, the Field Guide to Pleistocene Hookups, which is another way of putting that all of these ancient people seem to have occasionally interbred with each other, and when they came into contact, mixed with each other. And again, it's the same story of populations expanding, it's growing, it's entering a new area, it mixes a little bit with the inhabitants. Subsequent populations have some small fraction of the genomes of these ancient people still in them. And then maybe later on, somebody else comes along and covers up the evidence. Well, I want to talk about the covering up the evidence aspect, because we now have DNA from Neanderthals. This is a mitochondrial phylogeny of Neanderthals, and lots of different Neanderthal sites here. And what I've highlighted, this is from a paper by Love Dahlen and colleagues from Uppsala. And this paper shows that a lot of Neanderthal sequences fall into a fairly closely related clade, and a lot of them fall into a sort of a broader group of clades. These are all the young sites in Western Europe. These are all the other Neanderthals. And here we're sampling across time. So these are all, as you see, earlier sites out in Western Europe and later sites here in Asia. And here you've got a lot of later sites in Western Europe. So it's like something happened with Neanderthals. A group of them picked up and spread across Western Europe. We once would have predicted that the earlier Neanderthals in Western Europe were the ancestors of the later Neanderthals in Western Europe. We would have interpreted that as a straight line. Now we know from genetics that it is a more complicated line. Probably there are lines of descent from earlier to later in Western Europe, but there are also significant lines of migration. Nowhere is this more true than at Sima De Los Huesos. Sima De Los Huesos is one of the most famous fossil hominin sites in the world. It has the largest single collection of hominin fossils found anywhere. I know this because now I'm working on the second largest collection found anywhere. So we're aiming for this. We're not going to get there. There's at least 33 individuals here, and their skeletal representation is pretty complete. It's here in northern Spain. And it dates to 400,000 years ago. This is Juan Luis Arzuaga, who's been working at the site since the early 1980s and is responsible for excavating most of these specimens, actually. Here's the family of the Sima De Los Huesos hominins. People debated these for years. They're 400,000 years old, which is earlier than we find evidence of real Neanderthals, all the Neanderthal features Neanderthals. And so there's this disagreement. Are these early Neanderthals who hadn't yet evolved Neanderthal features, or do they represent an ancestral population that might be more primitive and shared with the ancestors of modern humans? Maybe these are something like the common ancestors of modern humans, sub-Saharan Africans, and Neanderthals. Well, it turns out from their genetics that this model is what accounts for them. These are early members of the Neanderthal lineage. But I'll tell you that, and they postdate the separation of Neanderthals here and Denisovans. These fossils are 400,000 years old. We infer that these events must have happened earlier than that. And probably for the ancestors of sub-Saharan Africans today and Neanderthals and Denisovans, we're looking at a date that's something like 700,000 years ago. So this is the time frame we're talking about. That means that the population that contributed to Denisovans must go back yet further in time than that. But as we look at populations like this, the Sima De Los Huesos mitochondrial DNA is more similar to the Denisovan mitochondrial DNA than it is to other Neanderthals. These populations, we can conclude, are successively changing their variation because of successive migrations and mixtures that involve groups of people. So these are not static populations that are establishing themselves and remaining as vicariant populations for hundreds of thousands of years. They're actually dynamic populations that are continually being replenished and are evolving their variation over time from some source populations. Of course, in the paleontological record, we're not necessarily sampling the sources. We sample probably mostly sinks. So let me turn to a final topic, and that is these crania. This is a Neanderthal, the most famous Neanderthal of all, La Chapelle-aux-Sands, the old man of La Chapelle. And this is a modern human, the most famous early modern human skull of all, Cro-Magnon. You guys have heard of Cro-Magnons. This is modern humans. Up until yesterday, up until 10 years ago, we used to think of the Cro-Magnons who followed the Neanderthals in Europe as the ancestors of today's Europeans, right? We know what happened. The Neanderthals were there. They became extinct. Cro-Magnons spread. They took their place. Today's Europeans come from them. And it wasn't nonsense that we thought that, right? If you look at this skull, you can see this high nasal angle and this face with a really pronounced canine fossa there, this divot. It's got sort of a somewhat sloping but much more vertical forehead than the Neanderthal. You look at this skull and you say, wow, this is a modern human. And moreover, a forensic anthropologist looking at this would say, well, it's sort of European looking. It doesn't really look like an African skull. It doesn't really look like an Asian skull. It's a European. This must be where today's Europeans came from. When they looked at the skulls from this site, from Mount Carmel, I showed you at the beginning, and this site, Amud. Amud means pillar. It's got this pillar of rock outside of it. This is just north of the Sea of Galilee. Here's the Amud skull. Here's one of the Mount Carmel skulls. This is School 5. This is actually at Harvard. We do have an American early modern human. It's just a short drive away, guys. So Neanderthal, modern human, we looked at these and said, well, these must be the proto-Cro-Magnons. This must be where the modern humans came from. They emerged from Africa. They're here in the Levant 100,000 years ago. They ultimately get to Europe to replace the Neanderthals after 40,000 years ago. Tidy story. That tidy story is screwed up by this specimen. This is from a place called Oase. Oase is in Romania. And Oase is a cave. And this is a place where, in order to get to where the skull and the mandible are, you have to swim underwater in the cave and come up. So you have to have diving equipment to go in there and work. 2 specimens have been found there, 2 different individuals. This individual's genetics are more Neanderthal than any living human. He's got between 6 and 10% Neanderthal in his ancestry. And his Neanderthal, unlike yours, comes in big chunks. Big, contiguous chromosome chunks of Neanderthal. Oase 2, this skull, had a Neanderthal great-great-great-grandparent. Oase walked among the Neanderthals. And this skull, the earliest modern European, around 45,000 years old, this skull marks the time that those populations are in contact. Now, there's a problem with that. If he's in a population where he could be 10% Neanderthal, why isn't anybody today 10% Neanderthal? Where did it go? And if he's in a population where his great-great-great-grandparent was a Neanderthal, why is it that everybody today looks like our Neanderthal ancestry came in 70,000 years ago? There's an answer to this, and it's an answer that we guessed at from morphology. Here's a feature of the mandible, this mandibular foramen. Neanderthals typically have a form that's like this, that's just bridged over. We call it the horizontal oval. And when we look at its frequency over time, Neanderthals have this really commonly, but the first people after Neanderthals had it a lot too. And it declines continually up to the present. Here's another feature of Neanderthals. You see, it's not an instantaneous drop, it's actually a decline. What's going on there? We now know what's going on. When we look at Europe and look at the genetics of people across Europe, their genetics today are a very good marker of where they live. If you sample people who have 4 grandparents from Paris, 4 grandparents from Sofia, 4 grandparents from Madrid, right? This is what you get. Plot out their genetic variability, and it plots on 2 axes, principal component 1, principal component 2, and all you have to do is to overlay the map of Europe on this to realize that, oh, here's Spain, here's Portugal, here's France, here's Switzerland, here's Belgium, here's Deutschland, here's Denmark. It's exactly where you think it should be, right? Humans' variation today plots with the map, which is why you can send your DNA to 23andMe and they'll tell you where your ancestors came from. But when we look at ancient people in Europe, here's that same map, stretched a bit because some West Asian populations are in it. When we look at Neolithic people in Europe, when we look at Pleistocene people, hunter-gatherers in Europe, we find that they map completely differently. The ancient DNA from these people is no longer represented in today's populations. When we look at the mitochondrial sequences in early farmers versus the hunter-gatherers, these are the Cro-Magnons. These are people that followed the Neanderthals. You see that, wow, this is a complete genetic turnover. The early farmers of Europe have something like 5 or 10% of the genetics of the people that lived before them. Europe, according to ancient DNA, is a totally changed continent multiple times. So when we look at Europeans today, we're looking at the legacy of ancient populations from sources that mix together, and those ancient sequences come from populations that don't exist anymore. This is what we're getting from ancient DNA. And that's sort of the story that I want to end on, because when we look at today's Neanderthal ancestry, we're looking at something that's been filtered through 40,000 years of population mixtures and movements. It's really great to send your DNA to 23andMe. It's on the personal genomics front, right? It's really great to send in your DNA and they'll tell you how Neanderthal you are. But what they're telling you when they do it is how much you're not like the people that came from the same place you've come from, and how much genetic transitions of the past have influenced today's genetics. We're looking again and again and again at massive genetic events that reflect population growth, dispersal, cultural changes, all of those things that we can look around the world today and see happening have massively affected our populations prehistorically. And that's a much more interesting history, I think, than we expected to have found. All right, thank you, everybody. I'm happy to take some questions. But I also know that some people probably have to leave, so if you have to leave, don't feel bad. But I'll be happy to stay and answer some questions as well. Yeah?
So you had for Devonian,
For
the
Denisovin.
Denisovans.
Yes.
You had 2 lineages that were quite separate.
Yeah.
Same site, different sites?
So what's going on is that we have Denisovan DNA from one site, and we have traces of a Denisovan-like population that intergressed, that put genetics in today's people. those are, we infer, fairly different. More different, approximately as different as the most divergent living people are from each other. So it's like the Denisovans of the past were a structured population which was maybe as diverse as today's African population. And the part of it that interbred with modern humans was distinct in some ways from the part that we've sampled from ancient DNA. Yeah?
Given how much change that you reported at the beginning of your presentation, I'm going to guess it's going to continue to accelerate. Tell me a little bit about the prospects for deepening the reach of DNA to reach these ghost populations or other unknowns.
We're limited in part by preservation, in the sense that there are only a very small number of sites where the preservation is ideal. At most sites, the preservation is poor. We've gotten DNA out of a large number of Neanderthals, but for the most part, it's not economical with today's technology to do better. So we know there's DNA there, but it's highly contaminated, it's highly broken up into short fragments, and it becomes possible to reconstruct a mitochondrial genome because there's 1,000 copies of the mitochondrial DNA for every nuclear genome. But beyond that, it's cost prohibitive to go further. There's DNA there. And with better technology, we could probably do better. The Sima De Los Huesos DNA, which is 400,000 years old, is predominantly broken into fragment lengths of less than 40 base pairs and mostly clustered 20 to 30 base pairs. So you're looking at really the limits of being able to differentiate a human-like fragment from, even microbial fragments start to fall into that range. So I think that's probably near the upper limit in terms of time depth, and it is certainly beyond the upper limit in terms of economics at the moment, although as technology improves, the economic limit will become probably less important. Yeah?
So I wonder if you'd care to speculate on what is the underlying cause of all of these turnovers in European populations over the last 20,000 years?
It's a really interesting question, and one that gets to where my lab is really going, because the question is, what accounts for all this turnover, particularly in Europe, right? Partly, it's got to be selection driven. Demographic expansion in recent human populations is, there's culture that contributes to why some populations grow and are better at growth. But over this kind of time, it's got cultural traits you can learn, right? We used to think that maybe people picked up farming technology and it spread gradually across Europe as some sort of diffusion, We now know that actually farmers came. And people can learn how to farm. And ultimately, those people did mix a bit, but there seems to have been a sort of a persistence of. This seems to have been true in the Bronze Age. You've got these people that move through Europe, and they're warriors, and they probably kill a lot of people. And they take over other places, and the genetics of the European population is different after that. What drives that? Well, actually, people showed up and killed people. But also, this is when some major selective changes are happening in the gene pool of these people. You've got things like lactase persistence increasing in frequency, so suddenly you can grow your population faster if you're dairying than you would have been able to do otherwise. So I think it's a combination of cultural phenomena which are increasingly sort of demographically harmful, and selection and coevolution, making people better and better suited to these cultural shifts. Yeah.
So how do you get selection in these kind of small, dispersed populations? Isn't that the ideal situation for drift, for instance?
This is a great question, right? Because I used to think a lot about drift and gene flow. And there's a problem, right? In the old days, you had the problem of how do we get Neanderthal genetics into us? If you thought that it was there, right? Many people doubted it was there. Well, it's just gone. I said, well, it doesn't seem very plausible it's gone. And we've got these morphological features that seem to show some gene flow. But how does that actually happen? Because when you're in small dispersed populations and you're spreading genes, even if the gene is driven by selection, it takes a darn long time to spread, to propagate, you know? We used to do estimates of this, and you could show that if something was selected and its advantage is 1%, you could move it across the old world in 800,000 years. And it's like, that can't work, right? That's not what we're looking at. I think what we're looking at is that growth happens faster than dispersal. And at a certain phase, once your population's gotten big enough, you just have sort of this hopscotch sort of dispersal. When farmers enter Europe, it's happening because people, it's not happening because people are moving like, next door. It's because people, eventually their population gets big enough and they move 40 miles down the road. Or they move 400 miles down the road. And you've got this sort of hopscotch effect. It's much more like, if you want to think what it's like, it's not like beach mice, where you're looking at selection in a gradient. It's more like tamarisk, where you're looking at this sort of invasive species that hops to different river valleys, and there it grows. And I think that's what's going on with these populations. I think the demography is driving it. The advantage of that is that if you have a series of steps where the population really is growing, every one of those instances of population growth increases the fixation probability of anything adaptive. So it's like, that's the only way adaptation can happen in a population where some parts of it are doing that, because that's every advantageous thing that's there has a better chance of making it. Yeah.
Can you tell us anything about changes in Neanderthal populations over the extent of that they were alive? I mean,
yeah.
either some degree of biological adaptation, or is there any evidence of cultural change?
It's clear that much of the interesting part of the Neanderthal record, the cultural stuff, is late. It is not evident yet whether that's because our record is better late, which it is, or whether they really got more interesting later. And this is a great question for us because, of course, it comes to the issue of, were those behavioral features that make later Neanderthals interesting something that came from our distant common ancestors with them 800,000 years ago, or something like that? Or is this a case of convergence or cultural transfer, some sort of communication? The eagle talons we now have for more than 120,000 years ago. So that suggests maybe if we look hard enough, we'll find more interesting things early. So that's a possibility. There were different morphological types of Neanderthals. And as I look at them today, you look at Levantine Neanderthals, the stereotype of Neanderthals is that they're sort of short, they're thick, they've got relatively short limbs, they're cold adapted. In Israel, they never were. In Israel, the Neanderthals are tall, and they have cranial features that connect them to European Neanderthals, but they're not profoundly European looking. The earlier Neanderthals, like those from Krapina, I say early, right? Now we have to include Sima De Los Huesos. They have very few of the classic Neanderthal configuration. It's clear that that population evolved a lot. Krapina is, in many ways, a less extremely morphologically differentiated population compared to modern humans, and it's 120,000 years old, than 45, 50,000-year-old Neanderthals were. The classic French Neanderthals are the most weird in that sense. So yeah, there's a lot of information there in the morphological record. That information is not yet there for us in the genetic record. I can't yet talk about gene frequencies over space, except for mitochondrial frequencies over space. All right, I'll take one more, yeah.
Have you looked into how, say, plate tectonics has played a role in evolution?
In this section of our evolution, very little. But what has played a big role is sea level. As sea level today's sea level is very high. It's a high stand compared to what was true for most of our evolutionary history. Mostly, we have sea levels that are 100 meters or more below where they are now. And so there are huge areas of some parts of the world that are exposed in the past that today are underwater. we actually have fossil evidence from some of those places where people dredging the bottom for gravel have found fossil hominins. So there's evidence of habitation there. They find stone tools quite a lot. And we're doing now some parts of the world, some underwater archaeology to investigate those past landscapes. All right, thank you again, everyone. Thank you so much.